Good morning, everyone.
Morning, sir.
Okay, so I'll start the class. So today we are going to discuss cholesterol synthesis and regulation. Okay, so we'll start with cholesterol. What is cholesterol? Okay, what is the structure of cholesterol, which we already saw in lipid chemistry, right? Then how we'll see how the cholesterol is synthesized. What are the sources of cholesterol? Then regulation of cholesterol synthesis, and what happens if the cholesterol level increases or decreases in the body, and then some drugs used against it. Okay, so as I already told in lipid chemistry class in classification that cholesterol is the most important sterol, okay, in our body. Okay, so cholesterol is found in different tissues also, and in the plasma also, in circulation also. And now this cholesterol can be present in two forms mainly. It can either be present as free cholesterol, or it can be present as cholesterol ester. And the carrier molecule which transports cholesterol in the plasma are known as lipoprotein. In the next class, we will discuss about lipoprotein. There are different types of lipoprotein: LDL, HDL, VLDL. You might have heard also. So, plasma low density lipoprotein. This is the major molecule for uptake of cholesterol from the basically from the liver, and it gives cholesterol and cholesterol ester to the tissues. This LDL mainly gives to different tissues and different organs of the body for synthesis of different products. So major products are like bile, then steroid hormones and all. Vitamin D is also synthesized from cholesterol, and HDL cholesterol takes up extra free cholesterol from the circulation and gives to the liver for its metabolism. And because this HDL is giving back the cholesterol back to the liver, this is known as reverse cholesterol transport. Because if more and more cholesterol gets deposited in different various organs apart from liver, generally that is not good for body. uh, it has bad effect, and we'll see how in the lipoprotein class. And so that's why generally you would have heard that LDL is a bad cholesterol, but HDL, because it takes up excess cholesterol from HDL takes up excess cholesterol from different tissues and it gives it back to the liver for its metabolism. That's why it is known as good cholesterol. A structure of cholesterol, we saw that there were four rings, right? You'll see here there are four rings. Okay, then there is one OH group and methyl group attached to the to the 10th carbon and 13th carbon. This 19 and 20 18 is the methyl group. This you can see the structure in the book also. So the structure you should just know schematically like this. Okay, how many carbon are there? Then how many side chain? How many carbon present in the side chain? Then where is the methyl group attached? And where is the hydroxyl group attached? Now functions of cholesterol. This is important. This can come in your exam for 2-3 marks. So this is the most abundant and most important sterol in the body. Okay, and its role are, it is a major constituent of plasma membrane, which you have read in your first few classes only. Is a major constituent of plasma membrane and plasma lipoprotein. Lipoprotein are the carrier of cholesterol, cholesterol ester, triacylglycerol. So basically, lipid transport, lipoprotein is very important and is a major component. And the role of cholesterol in plasma membrane also you would have read that it gives the rigidity and fluidity of the membrane. It depends on the cholesterol level. Cholesterol is also the precursor of bile salts. Bile salts and bile acids uh, are formed from cholesterol. So you have to homework, basically a small homework. You have to know the names of different bile acids and different bile salts. Primary bile acids, secondary bile acids, and bile salts. So you have to go back and read just the names. Then next, cholesterol, it is the precursor of steroid hormones. Steroid hormones are which are adrenocortical hormones, sex hormones, placental hormones. So another homework is you have to find out what are the adrenocortical hormones. You would have heard only like cortisol, then aldosterone and all. So you have to go back and read what are the adrenocortical hormones, names only, sex hormones and placental hormones. So these two you have to know. They might come in your exam. Now third is cholesterol is also a precursor of vitamin D. Other thing no need to know. So precursor of bile salts and bile acids, precursor of steroid hormones, which includes all these. You have to know the names. Then precursor of vitamin D. So vitamin D we might have separate class either I will take or from in nutrition they will be taking. And this cholesterol is required for nerve transmission also. So basically cholesterol is widely distributed. Basically most of the cell present in the body can form cholesterol. So what are the sources? Sources of cholesterol, three types. Either you take in diet, or de novo synthesis means completely new. It forms from acetyl coenzyme A or from the hydrolysis of cholesterol ester. If cholesterol ester breaks down, then also cholesterol can be formed. So more than half, almost 60% of the body cholesterol is synthesized inside the body. Okay, the rest we take from the diet. And liver and intestine are the most important like organs where basically cholesterol is synthesized, 10% each. And all tissues which contains nucleus can produce cholesterol, and the synthesis mainly happens, what are the place where the cholesterol synthesis happens is cytosol and endoplasmic reticulum. So site also you have to know for everything, like for fatty acid oxidation, fatty acid synthesis, like that cholesterol synthesis also mainly happens in cytosol and endoplasmic reticulum. So sources are diet, new synthesis inside the body, de novo synthesis, and hydrolysis of cholesterol ester. Okay, major organ are liver and intestine, but all the nucleated cell in the body can generally produce cholesterol. So steps. So it's a very complex process, but I'll tell you what all terms you have to know and what all step you have to remember. Otherwise, it is a very complex. So what is the precursor molecule? Acetyl coenzyme A. Acetyl coenzyme is the precursor. So this you have to know. And all the carbon atoms come from acetyl coenzyme. The major steps are three. Three stages are there. In each stage, there are many steps present. So in first stage, the synthesis of isopentenyl pyrophosphate. So basically, from acetyl coenzyme A, isoprene forms, and from isoprene, isopentyl pyrophosphate forms. So in first stage, you have to know these three terms only. When you write in flowchart, you will write acetyl coenzyme A, then arrow, then you will write isoprene, then you will write isopentenyl pyrophosphate. That's it. From isopentyl pyrophosphate, you directly will write esqualene. Okay, I'll tell you the number of carbon atom in each also, and you have to write carbon number also. And in third stage, you'll write from esqualene cholesterol is formed. Okay, so if the mostly I highly doubt that synthesis of cholesterol will come, but if you write this much is okay. But at each term, you have to write how many carbon is present in these molecules: acetyl coenzyme A, isoprene, isopentyl pyrophosphate, then esqualene and cholesterol. So from acetyl coenzyme A to isoprene, generally you have to know a little bit detail. I'll tell you. So first stage is the synthesis of cholesterol with the formation of isopentyl pyrophosphate from acetyl coenzyme A. So understood? So how that reaction proceeds, we'll see here. Two molecule of acetyl coenzyme A condenses with the help of enzymes thiolase. Where you saw this thiolase enzyme in ketone body synthesis also. I had told that initial pathway is similar in ketone body and cholesterol synthesis. Now acetoacetyl coenzyme A forms in ketone body also, right? From acetoacetyl coenzyme A, HMG coenzyme A synthase. This enzyme was also there in ketone body synthesis if you go back and read. So it will form HMG coenzyme A. Then this HMG coenzyme A will form mevalonate with the help of enzyme HMG coenzyme A reductase. So this is the regulating step. So you have to know this is the most important enzyme in the cholesterol biosynthesis. So most of the drugs also, or the feedback inhibition, all the regulation of cholesterol synthesis happens at this stage. Acetyl coenzyme A to acetoacetyl coenzyme A, thiolase enzyme. Acetoacetyl coenzyme A forms HMG coenzyme A with the help of HMG coenzyme A synthase, and HMG coenzyme A gets converted to mevalonate with the help of enzyme HMG coenzyme A reductase. And this is the committed step. Once mevalonate has formed, it will this acetyl coenzyme A will form cholesterol. So the regulation we will see. Normally, if most of the drug which acts against this enzyme also, if cholesterol excess is there, we give, and even in the feedback inhibition by the cholesterol is the end product. By feedback inhibition, it inhibits HMG coenzyme reductase. Once mevalonate has formed, mevalonate don't see these things. Just see that mevalonate forms isopentyl pyrophosphate. Okay, that's it. So acetyl coenzyme A, with the help of those enzymes, it is forming mevalonate. From mevalonate, it is forming isopentyl pyrophosphate, and this requires ATP. So you have to go back and see in book and write only the outline what I am telling you. Don't you can make notes easy. Okay, and from isopentyl pyrophosphate forms squalene. Isopentyl pyrophosphate, pentyl, so you can understand, right? Pentenyl. So five now, so it's a five carbon. So this six molecule of isopentenyl pyrophosphate will form squalene. Okay, so if 5, C10, 15, ultimately it will form 30. So if you directly write isopentenyl pyrophosphate to squalene. So isopentenyl pyrophosphate 5 carbon molecule, and right into 6 molecule will combine together and form squalene. No need to see this, and only in this step you can know that NADPH is required. Where else NADPH was required? NADPH was required in fatty acid biosynthesis also, if you remember. From squalene, once squalene has formed, it will form lanosterol, and lanosterol will form cholesterol. So not go into the detail. So basically, what is the step? Acetyl coenzyme A. Acetyl coenzyme A ultimately forms mevalonate. Mevalonate will ultimately form isopentenyl pyrophosphate, that will form squalene. 6 molecule of isopentenyl pyrophosphate, and squalene will form lanosterol. Lanosterol will form cholesterol. So that's it. Now regulation. I told you the committed step is the formation of mevalonate from HMG coenzyme A. The enzyme required for conversion of HMG coenzyme A to mevalonate is HMG coenzyme A reductase. So here only the regulation will happen. Now regulation, this is important question. This might come in your exam. How cholesterol synthesis or level in the body is regulated. So the 4 mechanism works generally here. HMG coenzyme A. First is competitive inhibition. Then second is feedback inhibition. If enzyme is over, you will be knowing what is competitive inhibition, what is feedback inhibition. Competitive inhibition, basically the enzyme required, it has some similar molecule to that, and that enzyme will go, that molecule will go and inhibit the action of the enzyme. Okay. Feedback inhibition, the product, either the immediate product of the enzyme or the end product of the pathway can inhibit the enzyme required. Okay. Third is covalent modification. So this is basically some hormones either activate or inactivate the enzyme by phosphorylation and dephosphorylation. If you, I don't know whether they taught you in enzymes or not, but phosphorylation and dephosphorylation is a process by which covalent modification happen and enzyme either gets activated or deactivated. So this is basically modulated by some hormones. Next is sterol mediated regulation of transcription. So this is the regulation at the transcription level, whether the HMG coenzyme reductase enzyme will form or not form. If transcription happens, then ultimately this protein will form from the gene. Enzyme is basically protein, right? So that protein has to form from gene. So there the gene can be upregulated, downregulated, and it will lead to increased production or decreased production of HMG coenzyme reductase. If there is increased production of HMG coenzyme reductase, there will be more and more cholesterol synthesis. So these are the initial steps: competitive inhibition and feedback inhibition. Then in second step, hormones have the role in modifying the structure basically of the enzyme by phosphorylation and dephosphorylation, and if further inhibition or activation is required, then that is done at the transcription level. Okay, so we'll go one by one, little we'll see. So competitive inhibition, normally these are the drugs, competitive inhibitors are the drugs we use to decrease cholesterol level in the body. Okay, so what it does, it inhibits the activity of HMG coenzyme A competitively. So it is a competitive inhibitor. So what are those drugs? Those drugs are known as statins. So you would have heard, or someone normally, if they have elderly people at home, they would have seen that they take normally atorvastatin or rosuvastatin or lovastatin. So statins act by inhibiting the activity of HMG coenzyme A reductase by competitive inhibition. That's it. So these are the drugs given to reduce cholesterol level in the body. Statins: atorvastatin, rosuvastatin, lovastatin. Just remember a few names. Second is feedback inhibition. So the HMG coenzyme A reductase forms what? Mevalonate from the HMG coenzyme A, and ultimately it leads to formation of cholesterol. So cholesterol and mevalonate all are product of this, ultimately product of what? The activity of HMG coenzyme A. So these products, basically mevalonate and cholesterol, inhibit the activity of HMG coenzyme A by feedback inhibition. More and more product has formed, so the activity of enzyme is not required anymore, right? So that's why it inhibits the activity of HMG coenzyme A reductase. Now covalent modification. So the phosphorylation of the HMG coenzyme A reductase decreases its activity. Now glucagon favors phosphorylation. Basically, by now you must be knowing that I have discussed many times that insulin is an anabolic hormone. Anabolic means what? It forms bigger molecules. And whatever you eat, smaller molecules in the in your diet. Normally, insulin will try to make a complex molecule from that and store it. Okay, so that's why insulin is anabolic molecule hormone, and glucagon breaks down the complex molecule in the body to a smaller molecule and increases the level of that smaller molecule in the circulation. So glucagon is known as catabolic hormone. So that's why it will not favor the formation of cholesterol. It is a complex molecule. So glucagon will phosphorylate basically. It will phosphorylate the enzyme. Which enzyme? HMG coenzyme reductase. So in phosphorylated state, HMG coenzyme reductase activity is reduced. Hence the rate of cholesterol synthesis will decrease because of glucagon. In contrast to glucagon, insulin will activate HMG coenzyme A by dephosphorylating. So basically, insulin is favoring the formation of HMG coenzyme A, which will result in more and more production of cholesterol and storage of cholesterol. Another thing important, I told you that ATP is required, right? So ATP is required for the formation of isopentenyl pyrophosphate from mevalonate. So cholesterol synthesis will decrease or gets inhibited if ATP level in the body decreases. So covalent modification, the role of hormones mainly. Which hormone? Glucagon and insulin. Glucagon will decrease the rate of cholesterol synthesis, and insulin favors the formation of cholesterol by activating HMG coenzyme reductase. So till now we saw what are the regulations. Competitive inhibition by drugs. The group of drug is known as statin. Okay, and it inhibits competitively HMG coenzyme A. Second is feedback inhibition. The product of the HMG coenzyme reductase, mevalonate and cholesterol, inhibits the activity of HMG coenzyme reductase. Third is covalent modification. So some hormones either favor or inhibit the formation of cholesterol. Next one is sterol mediated regulation of transcription. This is little complex, but see, you'll be able to understand. So the synthesis of cholesterol is also regulated by amount of cholesterol taken up by cell during lipoprotein metabolism, right? So basically during lipoprotein metabolism, if more and more cholesterol is taken up by the cell inside, so then the synthesis of cholesterol will not happen because already so much cholesterol will be present inside the cell. Chylomicron, this we'll see in detail, these two. Basically, these 2 points you'll see in the details in lipoprotein class, next classes. So chylomicron remnant internalized. Chylomicron is a type of lipoprotein we'll see. The remnant of the chylomicron internalized by liver cell. So these remnants basically just for now know that chylomicron is internalized by liver cell. It's taken up by the liver cells, and low density lipoprotein internalized by liver cells and peripheral tissue. So chylomicron is normally taken up by liver cells, and LDL is taken up by liver cells also and peripheral tissue also. These provide cholesterol. Okay, these provide cholesterol to the cells either in the liver or in the peripheral tissue. So what they will do, because it provides cholesterol to the cells, it causes decrease in the transcription of HMG coenzyme reductase gene, because already so much cholesterol has come inside the cells of the liver and the peripheral tissue. So this increased concentration of cholesterol inside the cell will decrease the transcription at the transcription level basically. So the transcription of HMG coenzyme reductase gene is decreased. If transcription is not happening, what will happen? What level transcription happen? Transcription means from DNA to RNA, right? So mRNA, messenger RNA, will not form for this HMG coenzyme, and that will not form. Ultimately, then the protein, which is HMG coenzyme reductase, will not form, leading to a decrease in cholesterol synthesis. So understand, if more and more chylomicron LDLs is present in circulation, that will be internalized by liver cells and different peripheral tissues. That cholesterol, increased level of cholesterol inside the cell, will decrease the transcription of HMG coenzyme reductase gene, leading to decreased synthesis of cholesterol. Now the rate of synthesis of reductase, HMG coenzyme reductase mRNA, is controlled by, now how it is controlled, how this transcription is decreased. So that you have to know that the synthesis, transcription means the formation of HMG coenzyme reductase mRNA from HMG coenzyme reductase gene. From DNA to RNA is the transcription, and from RNA to protein formation is translation. I think you all must be knowing that. So this decrease in rate of synthesis of mRNA, reductase mRNA from reductase gene, is controlled by a protein known as SREBP, sterol regulatory element binding protein. Sterol regulatory element binding protein. Okay, so go back and read. So you'll remember SREBP. So how it happens basically? Now this transcription factor SREBP binds to short DNA segment called sterol regulatory element. Okay, so in its inactive state, this transcription factor SREBP is anchored to the endoplasmic reticulum. So this is when it is inactive. This protein SREBP, this is a regulatory protein which is also known as transcription factor, because this is required for increased transcription. So when it is inactive, where it is there present, it is present in the membrane of the endoplasmic reticulum and nucleus. So when it will get activated, when cholesterol level will decrease, right? When cholesterol level inside the cells decreases, this SREBP, which is the transcription factor, is released from the membrane of the nucleus and endoplasmic reticulum. Then this SREBP will go inside the nucleus and bind to SRE, sterol regulatory element. SRE of this gene. So HMG coenzyme reductase gene will be present in the DNA. So beside that gene only, this element is present. SRE, sterol regulatory element. This sterol regulatory element will bind with the SREBP, which is released from the membrane of the nucleus and endoplasmic reticulum when cholesterol level decreases. So once it binds to SRE, this leads to increased transcription of HMG coenzyme reductase gene. So increased transcription will do what? Basically, it will form more and more mRNA of HMG coenzyme reductase. So more and more mRNA of HMG coenzyme reductase is forming, then more and more HMG coenzyme reductase enzyme, which is a protein, will form, and more HMG coenzyme reductase enzyme will form, more and more cholesterol will form. So I will say, if anyone want to write, you can write. Basically, SREBP is a transcription factor, okay, which is required for the transcription of HMG coenzyme reductase gene. When generally, because of the excess chylomicron and VLDL and LDL, more and more cholesterol goes inside the cell, it inhibits transcription. Okay, but whenever the level of the cholesterol falls inside the cell, this SREBP transcription factor, which is a protein basically, is released from the nuclear and endoplasmic reticulum membrane. So once it is released, it goes inside the nucleus and it binds to a sterol regulatory element region of the DNA, which is beside the HMG coenzyme reductase gene. So once it binds, SREBP binds to SRE, the transcription of HMG coenzyme reductase gene increase. So that increase in transcription leads to more and more formation of mRNA of HMG coenzyme reductase. More mRNA of HMG coenzyme leads to more formation of HMG coenzyme reductase enzyme by the process of translation, and more HMG coenzyme reductase enzyme leads to increased formation of cholesterol. So basically, this is how the sterol mediated regulation of transcription of HMG coenzyme reductase enzyme happens. So when cholesterol level will rise, this SREBP is blocked, and whatever is present inside the nucleus that cannot go back and attach to the nuclear membrane, so those proteins will get degraded. You have to go back and read. It's simple only. You have to just know the sequences. Don't transport. We'll see the detail of transport in lipoprotein metabolism, but see the like little bit we'll see here. So generally, cholesterol is transported in plasma with the help of lipoproteins. Okay, so lipoprotein are the basically carrier. We'll see the structure also of the lipoprotein, what are the constituents of lipoprotein, everything in next few classes. And the highest lipoprotein are different types. You would have heard chylomicron, VLDL, IDL, LDL, HDL, and the maximum quantity of cholesterol is generally present in LDL, low density lipoproteins. Now the cholesterol ester which we take in diet. So in diet you can take a cholesterol in the form of cholesterol also, and some cholesterol comes in the form of cholesterol ester also. So this cholesterol ester is hydrolyzed to cholesterol in the intestine. Okay. So this first cholesterol ester will get hydrolyzed to cholesterol in the intestine, and this cholesterol which is now hydrolyzed from the cholesterol ester plus the cholesterol which you have taken in the food will get absorbed together in the intestine with cholesterol synthesized in the liver. So in intestine. So whatever you are taking in diet, that cholesterol also. Then I told that intestine is also a major organ of cholesterol synthesis. So the cholesterol which is synthesized de novo, new cholesterol which is synthesized in the intestine, all those hormones will get incorporated into chylomicron. Chylomicron is a type of lipoprotein, it is a type carrier molecule for cholesterol. So the cholesterol which you are taking in food and cholesterol which is getting synthesized in your intestine, all those cholesterol will go inside the chylomicron, and this chylomicron will carry the cholesterol from diet and intestine and deliver it to the liver. So for now you remember this much only: the cholesterol ester and cholesterol whatever we are eating plus new cholesterol synthesized in intestine gets incorporated into the chylomicron, and this chylomicron delivers these cholesterol into the liver. Okay, so how it is delivering, all those things we will see in next class. And most of the cholesterol secreted by liver in VLDL. So, and cholesterol secretes here, we saw that chylomicron is taking up cholesterol from the intestine to the liver. Now the cholesterol will be secreted from the liver also. So they will be in the form of VLDL, which will form IDL and which will form LDL sequentially, and this LDL will take up this cholesterol from the liver and deliver it to the mainly to the extrahepatic tissues. Okay, so chylomicron gives cholesterol from intestine to liver. From liver, this cholesterol comes out to the different other tissues, extrahepatic tissue, with the help of LDL. Remember this. And from extrahepatic tissue, cholesterol is given back to the liver with the help of HDL. We'll see this in details. And the LDL are taken up by the cell. So how this LDL gives the cholesterol and cholesterol ester to the extrahepatic tissue? The process is known as receptor mediated endocytosis. So generally cholesterol will go and bind to the receptor present on the cells, extrahepatic tissues known as LDL receptor, and then the whole receptor and LDL will be engulfed inside the cell. Okay, together, and then that cholesterol will go inside the cell and then it will be utilized for formation of vitamin D or steroid hormones and all. So LDL receptor mediates this endocytosis. Basically the LDL will bind to LDL receptor and then it will get endosesized. Basically endocytosis of the LDL receptor plus LDL is happening. After LDL binding to the LDL receptor, the ligand receptor complex, ligand is LDL and the receptor is LDL receptor, complex cluster plasma membrane in coated pit, which is then invaginates, forming coated vesicles. So basically this thing will get endocytosed. These coated vesicles are internalized and clathrin, the protein composing the lattice membrane coated pit, is removed. So we'll see in the next slide. So here, see, this is LDL receptor. It is coated with clathrin. Okay, this receptor is coated with clathrin. This cholesterol ester and cholesterol present in the LDL. This LDL is a what lipoprotein I told, right? Lipoprotein are different type. So generally in the membrane, lipoprotein will contain more and more proteins. We'll see those, what are those proteins also, and inside it will come cholesterol and cholesterol ester. So this LDL will be taken up in this clathrin coated pit, which is the receptor, and this whole clathrin coated pit plus this LDL which contains the cholesterol will get endosesized, which is known as internalization, and then it will be broken down. So the cholesterol which is released will be utilized for formation of different molecules in the cell, and this clathrin coated pit will go into the lysosome basically and gets degraded. So LDL binding, internalization, lysosomal hydrolysis. So these vesicles are now known as endosome. The whole vesicle which is containing the LDL receptor plus LDL is known as endosome, and this endosome will fuse with lysosome inside the cell. The LDL receptor containing membrane buds off and is recycled to the plasma membrane. So this will again, this LDL receptor after release from the lysosome will go and bind to the plasma membrane. Fusion of lysosome and endosome releases lysosomal proteases that degrade the apoprotein into amino acids. So this much no need to know, it's okay. But apoprotein is a part of lipoprotein, you know that. And lysosomal enzyme hydrolyze the cholesterol ester to free cholesterol. So now the cholesterol I told that can be present in cholesterol form also and cholesterol ester form also. So the enzymes which is present in lysosome will convert the cholesterol ester form to free cholesterol, and these free cholesterol will be utilized for the formation of steroid hormones and vitamin D and all. Free cholesterol is released in the cell cytoplasm, and this free cholesterol is then available to be used by the cell. So LDL in the extrahepatic tissue, LDL is taken up by the LDL receptor which is coated with clathrin. By the process of endocytosis, this whole receptor plus LDL gets endocytosed, and then the whole structure is known as endosome. This endosome will combine with lysosome. Lysosome will degrade the proteins present in the lipoprotein, and the receptor part will again go back to the plasma membrane, and the cholesterol and cholesterol ester will form the free cholesterol inside the cell, and this free cholesterol will be utilized by the cell in the formation of different molecules. And now the excess cholesterol, suppose after the whatever is cholesterol is used for the conversion into the steroid or vitamin D or other molecule, excess cholesterol can be re-esterified again with the help of enzyme acyl coenzyme cholesterol acyl transferase. So this will re-esterify the cholesterol with fatty acid. And free cholesterol affect cholesterol metabolism by inhibiting cholesterol biosynthesis. This we already saw, that by feedback inhibition, if excess cholesterol is present, then cholesterol biosynthesis will decrease in the body. Cholesterol inhibits the enzyme HMG coenzyme A, which is the rate limiting enzyme. This we already saw. And in addition, free cholesterol inhibits the synthesis of LDL receptor, thus limiting the amount of LDL that are taken up by the cell. So now if more and more cholesterol are present inside the cell, this will also inhibit the formation of LDL receptor, because then more and more LDL is not required in the cell. Already cholesterol is present. Now variation of cholesterol level in body. We have to know very outline. You know also something. Normal serum level is till 200. You remember till 200 only, not 220. Okay, 150 to 200. Normally till 200. And high cholesterol is normally found in diabetic patients. Okay, then nephrotic syndrome, then obstructive jaundice, then hypothyroidism. This you must have heard. Then familial hypercholesterolemia. This will be basically because of the genetic factors. So diabetes, nephrotic syndrome. Just know the few names when cholesterol level increases. Diabetes, nephrotic syndrome, obstructive jaundice. Nephrotic syndrome, why? Because in nephrotic syndrome more and more protein gets excreted, a smaller protein but bigger molecule, bigger proteins gets retained in the body, and more lipoprotein will form, and more cholesterol synthesis, circulation will happen in the body. So diabetes, hypothyroidism, nephrotic syndrome, obstructive jaundice. And obstructive jaundice, why? Because cholesterol degradation happen by formation of bile acids, and I told you to go back and read the names of bile acids. So cholesterol is degraded into the bile acids, and those bile acids and bile salt gets excreted in the intestine with the help of, and then it will get excreted, right? So but in obstructive jaundice, those bile acid formation decreases, right? It cannot come out in intestine, so more and more cholesterol circulation will be there in the body. So diabetes, nephrotic syndrome, obstructive jaundice, and hypothyroidism, remember that. High cholesterol level. Hypocholesterolemia, decreased cholesterol will be present in hyperthyroidism. Okay. Then malnutrition. Definitely you are taking less in food. Malabsorption. You are taking sufficient quantity of cholesterol in food, but it is not getting absorbed because of some intestinal problem. Then anemia. Then physiologically cholesterol level is less in children also, and the person on cholesterol lowering drug will have less cholesterol, right? Obviously. So this question can come, what will happen if there is a hypercholesterolemia, if increased cholesterol happen? And I think you all know that atherosclerosis happen. Atherosclerosis is basically what? Plaque formation in the tissue, in the blood vessels. So that plaque formation can be in the coronary arteries also, like the arteries around the heart, or it can be in peripheral tissue also. So this atherosclerosis is characterized by deposition of cholesterol and cholesterol ester. From where it is coming? It is coming from the increased circulation of cholesterol and cholesterol ester in the lipoprotein. So which lipoprotein normally causes circulation of cholesterol in the plasma? LDL, because it is taking cholesterol from the liver and it is giving to the extrahepatic tissue, and excess of this circulation is not good. So if excess happens, then the excess of the cholesterol and cholesterol ester from LDL generally will get deposited in the arterial wall, and it will form plaque like this. It will cause what will it cause? It will cause the narrowing of the artery, and ultimately it can lead to myocardial infarction also, right? Because if artery gets narrowed, then less and less oxygen will be delivered to the certain part of the heart muscle also, and sometimes it leads to myocardial infarction. So diseases in which prolonged elevated level of LDL, IDL, VLDL occurs in the body are often accompanied by premature and more severe atherosclerosis. So these things, increased circulation of LDL, VLDL, IDL, predisposes person in the formation of atherosclerosis plaque. Now, what are the drugs to reduce basically to reduce cholesterol level in the body, generally cholesterol or other lipids also. So those are known as hypolipidemic drug. Most important and most commonly used is statin. I told what are the statin? It is a competitive inhibitor of enzyme HMG coenzyme reductase. So what name you should know a few names of the statin also, like atorvastatin, rosuvastatin, lovastatin, all this. Then another group of drug is clofibrate. So clofibrate generally fibrate. So you remember clofibrate and gemfibrocil. Even if you not remember, just remember clofibrate. Clofibrate generally reduce plasma triacylglycerol. Statin generally reduce cholesterol level, and clofibrate reduces triacylglycerol level by decreasing secretion of triacylglycerol from the VLDL. So from VLDL is coming from the liver, right? Then only it will increase the level of the triacylglycerol and cholesterol increase in the circulation. So by decreasing the secretion of VLDL, clofibrate decreases the level of triacylglycerol. Just know the name statins, clofibrate. Then next is ezetimibe, and ezetimibe reduces cholesterol, blood cholesterol and other lipid level by inhibiting the absorption of cholesterol from intestine. So from intestine, the absorption of cholesterol and other lipids are decreased by ezetimibe. So every drug is working at a different level, right? Statin is reducing the formation of cholesterol by inhibiting HMG coenzyme reductase. Clofibrate is decreasing the secretion of VLDL from the liver, so thus it reduces triacylglycerol in circulation. And ezetimibe reduces the absorption of cholesterol and other lipids from the intestine. Next is bile acid sequestrants. Now this bile acid will sequestrants, basically these are resins which will combine with the bile acid in the intestine and promote their excretion. So it will combine, these bile acid sequestrants will combine with the bile acids and help in the excretion of these bile acids if through the intestine in the stool, right? So if bile acid decreases, then more and more cholesterol will get degraded and form bile acid. So that's how ultimately the cholesterol level also will reduce because of the increased degradation of cholesterol. And the other important drugs, hypolipidemic drugs, are omega-3 fatty acids, which reduces the cholesterol, cholesterol ester, and the saturated fatty acids. And another drug, niacin, is also hypolipidemic drug which reduces the free fatty acids. So you have to know the few names like statins, then clofibrate, ezetimibe, niacin, bile acid sequestrants, if question comes. Okay, so how you can reduce the cholesterol level by lifestyle modification. So generally the risk factor of increased cholesterol is high blood pressure, smoking and obesity. So by changing the lifestyle also you can reduce the cholesterol level in the body apart from using the drugs. Thank you. That's it for today. I'll be taking lipoprotein and its metabolism in the next few classes.
Thank you, sir.
Thank you, sir.
Okay.